The immunological structure of the skin. The skin isn't just a passive barrier, it's what we call a Grenzorgan, a border organ, meaning it forms a critical interface between the body and the external environment. Because of this position, it has its own immune capacity. The cutaneous immune system, which consists of both innate and adaptive, would be first discussed. The immune system must stay alert but avoid overreacting to harmless environmental factors to prevent chronic inflammation or autoimmune responses. So the second part to be described is homeostasis. And the third part to be discussed in here is the skin immunity in pathological signals. The skin's immune response is a combination of both innate and adaptive mechanisms. These two arms don't work in isolation, they interact closely to provide layered protection. This gives the immune system in the skin a dual nature. On the one hand, it has a protective role in a logical barrier. On the other hand, the system is also regulatory. Skin as target and source of immunity. Inflammatin from serum sickness showed immune complexes depositing in skin vessels demonstrate that the skin can be an immune target. Smallpox vaccination used the skin as an entry point for immune activation, highlighted skin as an active immunological organ. Contact dermatitis is a classic example of cell-mediated, type 4 hypersensitivity reaction, demonstrated that skin-resident lymphocytes can drive inflammation and memory responses. Antigen presentation role of Langerhans cells showing the skin's role in initiating adaptive immunity. Skin-associated lymphoid tissues. Langerhans cells in epidermis and dermal dendritic cells capture and process antigens that penetrate the skin barrier. After antigen uptake, they migrate to regional lymph nodes to present antigens to naive T cells, initiating adaptive immune response. Keratinocytes release cytokines like IL-1, TNF-alpha, IL-6, and chemokines that recruit and activate immune cells. Leukocytes in the skin, macrophages, T cells, neutrophils, also secrete cytokines that fine-tune APC function. The skin immune system. Skin is an organ capable of mounting immune responses. The innate immune. Innate immunity works by recognizing danger signals, either from invading microbes or from our own damaged cells, DAMPs. These signals are detected by pattern recognition receptors on skin cells and immune cells, allowing the skin to respond quickly without needing prior exposure. Adaptive immunity takes longer to develop, typically about 10 to 14 days after the first exposure to an antigen. Innate immune cells. Resident non-leukocytes such as keratinocytes, fibroblasts, sebocytes, endothelial cells and nerve cells, neutrophils, eosinophils, basophils are involved in innate immune cells. Other immune cells such as macrophages, mast cells, innate lymphoid cells, dendritic cells and NK cells are also involved in innate immunity. Innate immunity in skin. The skin expresses a wide variety of pattern recognition receptors, PRRs, that detect danger signals from pathogens or tissue injury. These receptors are found on keratinocytes, dendritic cells, macrophages, and other immune cells. PRRs recognize conserved molecular patterns rather than specific antigens, making them crucial for innate immune activation. Major class of PRRs are Toll-like receptors, NOD-like receptors, IgI-like receptors, and C-type lectin receptors. PRRs detect microbes, chemical haptens, and even UV-induced damage. Activation of these receptors triggers inflammatory cytokine production, example, IL-1, TNF-alpha, IFNs. Recruitment of immune cells to the site of insult and maturation of antigen-presenting cells, like Langerhans and dendritic cells. Once the skin detects injury, infection, or stress, it releases a wide range of soluble mediators, cytokines, chemokines, antimicrobial peptides, complement proteins, and enzymes, coordinate inflammation, immune cell recruitment, and tissue repair. Cytokines, master regulators of inflammation. TNF, potent pro-inflammatory cytokine, increases vascular permeability and recruits leukocytes. IL-1 beta, key in early inflammation, induces fever, activates endothelial cells, and promotes cytokine cascades. IL-6 drives acute phase response and promotes B-cell differentiation. IL-8, CXCL8 strong neutrophil chemoattractant, bridges cytokine and chemokine functions. IL-12 promotes Th1 differentiation and IFN gamma production, enhances cellular immunity. IL-10, anti-inflammatory cytokine that limits excessive immune reactions and promotes tissue repair. TGF-beta regulates wound healing, fibrosis, and immune tolerance in the skin. Chemokines immune cell trafficking signals. CXCL1 and CXCL8 recruit neutrophils to infected or injured skin. CCL3 attracts macrophages and T cells. CCL5 recruits T cells, eosinophils, and basophils associated with chronic inflammation. CCL20 recruits immature dendritic cells and memory T cells, key for skin immune surveillance. AMPs, antimicrobial peptides, direct microbial killers. Complement system, a rapid defense and inflammation amplifier. Key innate functions. Neutrophils, it is the first responders, phagocytosis, and extracellular traps. Eosinophils, anti-parasite defense by cationic proteins. Basophils and mast cells, histamine, IL-4, IL-13 allergy and inflammation. NK cells, recognize stressed or infected cells, kill via cytotoxicity. Innate lymphoid cells, ILCs, are the cytokine producers. Adaptive immune cells. Dendritic cells. T lymphocytes. Naive and memory subsets and tissue-resident memory T cells, TRM, are involved in adaptive immunity. Adaptive immunity in skin. Dendritic cells are key antigen-presenting cells. They capture, process, and present antigens to T cells to initiate adaptive immune responses. Langerhans cells, found in the epidermis, are a specialized subset of dendritic cells. They monitor the outermost layer for pathogens and can migrate to lymph nodes to activate T cells. In the dermis, there are other types of dendritic cells, notably CD1C+ and CD141+ DCs, which have distinct roles in antigen presentation and immune regulation. Moving to T lymphocytes, there are two main functional types. CD4+ helper T cells, which coordinate and regulate immune responses, and CD8+ cytotoxic T cells, which directly kill infected or damaged cells. These T cells exist as naive and memory subsets, previously activated and ready for faster response. Memory T cells persist in skin. Memory T cells has central and effector memory T cells. Effector memory T cells has recirculating TEM and tissue-resident memory T cells. Of particular importance in the skin are tissue-resident memory T cells, TRM. These remain in the tissue long-term and provide rapid, localized protection upon re-exposure to a pathogen, contributing to barrier immunity. Langerhans cells. Langerhans cells, LCs, are a specialized subset of dendritic cells found in the epidermis. They contain Birbeck granules, which are unique organelles marked by Langerin, CD207, important for antigen processing. LCs are versatile in antigen presentation. They can present both peptide antigens, typical protein fragments, and non-peptide antigens like lipids. In the steady state at rest, they help maintain immune tolerance by supporting regulatory T cell, TREG activation. This prevents unnecessary inflammation to harmless environmental antigens. Upon sensing danger, infection, injury, or stress, LCs become potent activators of CD4+ helper T cells and CD8+ cytotoxic T cells, initiating adaptive immune responses. This dual role highlights their importance in balancing tolerance and immunity in the skin. Skin immune system at homeostasis. Homeostasis is required to prevent exaggerated responses to harmless antigens, example, pollen, dander, food proteins. It also required in host defense reactions at a subclinical level without inflammation. Subclinical defense mechanisms. The skin employs constitutive antimicrobial mediators as a first line of defense against microbes. These include enzymes like RNases and proteases, which degrade microbial components. Antimicrobial peptides (AMPs), such as psoriasin S100A7, directly inhibit pathogens. For example, psoriasin is effective against E. coli. Commensal bacteria like Staphylococcus epidermidis play an active role by stimulating TC17 cells. TC17 cells produce IL-17, which acts on keratinocytes to induce further antimicrobial mediator production. This network helps maintain microbial balance on the skin, preventing overgrowth of harmful pathogens. Antigen presentation at steady state. Dendritic cells, DCs, constantly present antigens, but antigen presentation does not always trigger immune activation or sensitization. In the absence of danger signals like infection or tissue injury, DCs provide tolerizing or negative signals to T cells, promoting immune tolerance rather than inflammation. Langerhans cells, LCs, play a key role in this process. They were historically thought to be the main initiators of sensitization in the skin. However, more recent research shows that LCs often promote tolerance, helping prevent inappropriate immune responses to harmless antigens. This highlights the dual role of LCs and DCs in balancing immune surveillance with prevention of autoimmunity or allergy. Langerhans cells in homeostasis. Langerhans cells, LCs, are not static. They migrate to lymph nodes even in the absence of inflammation. During this migration, they carry antigens from the skin, such as melanosomes, pigment-containing organelles, and apoptotic bodies, cell debris. This constant trafficking of LCs and other dendritic cells helps maintain antigen-specific unresponsiveness, a form of immune tolerance. For example, in nickel allergy, this mechanism contributes to the generation of nickel-specific regulatory T cells, TREGs, which suppress inappropriate immune responses. So, most contact allergy to nickel exhibit without any clinical sign of nickel allergy.